The overall goal of this procedure is to visualize the movement of the actin binding protein in yeast that express a mutant actin isoform. This is accomplished by first making a plasmid encoding the actin interacting protein 1, or Aip1p gene tagged with the 3x green fluorescent protein fluorophore. The second step is to generate a yeast strain expressing only wild-type or mutant actin. Next, the GFP-tagged Aip1p plasmid is transformed into the wild-type or mutant yeast strain. The final step is to image and analyze the movement of the tagged Aip1p in the wild-type and mutant yeast cells. Ultimately, total internal reflection fluorescence or TIRF microscopy is used to show that the Aip1p moves slower in yeast expressing the R256H mutant actin than wild-type actin.
This method can help answer key questions in the cytoskeletal field, such as the relative temporal movements of binding partners. We first had the idea for this method when I discovered that, unlike the wild-type strain, deleting Aip1p from the mutant yeast strain was lethal.
The main advantage of this technique over existing methods such as confocal microscopy is the enhanced contrast and imaging speed. Generally, individuals new to this method may struggle to optimize the microscope settings for their individual experiment because of the numerous adjustments that can be made and the subjective nature of the image quality.
Begin this protocol with generation of the Aip1p GFP plasmid and mutant yeast strain as instructed in the text protocol. Digest the plasmid with a restriction enzyme to allow integration into the yeast chromosome. Culture the prepared S. cerevisiae.